The last talk will be given by Wan Kung Wong, who is a professor here at the Hong Kong University of Science and Technology and somebody who I've had the pleasure to meet and get to know as part of the organization of this conference. And there are a couple things that I think make Wan Kung unique. The first is his beautiful skin. He is interesting because he's not only a user of synthetic biology in the laboratory, but he's a user of his own product every day. And I think that shows. And I think it'll be a very interesting talk that he gives. Very few of us will actually apply what we produce onto our own basis.
Thank you, Chris. Good morning. Okay, my talk is cosmetic production in E. coli. As a matter of fact, cosmetic here may be misleading because I'm going to talk about how to make use of E. coli to produce epidermal growth factor. And as a matter of fact, epidermal growth factor in our laboratory has been used commercially as an active ingredient in cosmetics and also in skincare products. So the first part of my talk will be to introduce the system that we use to make large-scale production of epidermal growth factor. And later on, and then I'll talk about the applications of epidermal growth factor. Now I try to focus, of course, cosmetic, but how to get beautiful skin is very important. For example, healthy skin, So I'll also, I will try to focus my talk in using EGF to heal, as a killing agent to heal some of the hard to heal wounds, later on. Now, our group has been interested in epidermal growth factor, which is a small peptide, 53 amino acid, for many years. And as you know, that epidermal growth factor is important, and every one of us produce epidermal growth factor, which is important in the turnover of our skin, the upper layer, which is called epidermis of our skin. And as you know that EGF, epidermal growth factor, is found in many places in our body. For example, salivary glands. And as a matter of fact, all mammals produce epidermal growth factor, like cats, dogs. And that's why, when they have wounds, they lick, their wounds in order to improve healing. And also in our gut duodenum, for example. Now, since the discovery of epidermal growth factor in early 1960s, there have been a lot of research studies in the applications of epidermal growth factor. And there have been a lot of studies documenting the effects of it in wound healing, in particular burns, ulcers, for example, and also repair of our cornea damage, skin applications, as well as medical applications. Now, but if you go to the internet, and then you do a search, then you know that epidermal growth factor, the retail price of it is very expensive, easily amounts to 3,000 Hong Kong dollars per milligram. We have done some search, and then you can see that, many of the life science companies, they provide epidermal growth factor, but they are all very expensive in terms of multi-million dollars per gram. That's making it unaffordable, and it is not cost-effective to make use of it as an active ingredient, even though it is very useful medically. So one way to lower down the cost is, of course, to make use of genetic engineering. And our group has been very interested in, developing microbial systems to produce valuable proteins. In this case here, we have been interested in making use E. coli, Escherichia coli, as the host to produce EGF. But we try to use E. coli in an unconventional way. Rather than producing it inside the cell, we would like to express it as an extracellular product. I'll tell you why we would like to do that. And back to late 1980s, we started to develop this so-called extracellular production system, excretion, we call it. And our idea is that it is very important that, which is the point, we link the gene to a very strong, promoter, and of course, between the promoter, we make use of what we call the secretion signal. Now, this secretion signal has to be very effective in secreting the product to the periplasm. So we engineer what we call the excretion construct. And now let me use this diagram to explain our idea. The recombinant construct harboring the gene of interest inside the cell with the signal peptide or the leader sequence at the 5' end of the target gene. After expression, transcription and translation, then the leader sequence will guide the premature protein towards this inner membrane. As we know that E. coli has two membranes, the inner and the outer. So there are not too many proteins that could be transported to the outside part, the medium of the cell, naturally. So our idea was that we try to express the target protein as much as possible, very effectively. Hopefully, then the protein could be secreted to a large quantity, and this will form some sort of stress, and then forcing the cell to pump out the protein to the medium. Now, it works beautifully for actually EGF. So, in late 1980s, we obtained a worldwide patent on this system. And then we also, did a lot of research to try to see whether we could be able to get better or more effective production of foreign protein. For example, just recently, we find that we published in Protein Expression and Purification, we know that, if we could be able to control the passage of the secretion of the target protein to the periplasm through this so-called SCY, the SECD pathway, then we could be able to have more successful as well as effective production of the foreign protein. And so together with high density fermentation, then we have been able to demonstrate that using this approach, we could be able to express extracellular epidermal growth factor to an extent of 325 milligrams per liter. And that's so far the highest productivity, recorded in the literature. And as shown in this Comasi blue stain gel, you can see that HEGF is the major protein excreted to the supernatant. So these are all supernatant samples, and this is the marker. And that was proven by Western blot. And of course, since we have to concentrate EGF from such a large volume of culture, then we have to make use of large column. So in our lab, we have, commercial, semi-commercial scale purification system for the purification of EGF from the supernatant. And finally, we have to make use of HPLC to purify the product. Now, one important thing as you know that E. coli, the outer membrane, has what we call the endotoxin. Now, one nice thing about secretion and finally excretion of the product to the medium, it all happens naturally. We don't have to break the cell. So this will leave the endotoxin, the integrity of this outer membrane, intact. And also, because the protein is secreted or excreted to the medium, then it is already partially purified. And because of the more oxidizing environment in the medium, the product is biologically active. So, of course, we did a lot of characterization of the product excreted to the supernatant. For example, by running gel, when compared with commercial HEGF, basically there's no difference in terms of the mobility between the EGF commercially available and the one that prepared in our laboratory. And as what I said, our EGF is finally purified using HPLC, and you can see that the purity is very high. It is almost a single spike of protein. And we also, of course, concerns about the bioactivity. So when compared with a commercially available EGF obtained from Prepotec, then you can see that basically the EGF, the two EGFs, the one produced in our lab and the one that available commercially, they are equal potent, all right, equal potent. Now, the characterization of EGF, the properties are summarized the results are summarized in this table. And basically, the EGF is the same as what we produce in our body. And it is very pure, and it is fully bioactive. And of course, we're concerned about toxicity as well as whether it has it will cause any hypersensitivity. And we send our samples out and for testing, and basically the results are negative. Now, most importantly, the EGF we produce is now much cheaper than the commercially available EGF, almost 15 to 20 times cheaper than the commercial EGF. So now, of course, we have a more affordable way to produce EGF, then we always thought about how to commercialize our the product. So just recently, in year 2006, we have developed a biotech company called Ginvenate. So Ginvenate basically is comprised of three parties. BioClick Technology Limited, and that was founded by myself and one of my colleagues, and basically this company hosts the IPs, intellectual properties, and China Nansa Technology Enterprises, which is an investment company, pumping in money for us to develop our business. And most importantly, our university, the Research and Development Group Limited, is also a partner or shareholder of the company. Now that will facilitate the use of the laboratory facilities for us to produce EGF. That is very important. Otherwise, I mean, we could not be able to, produce the EGF cost effectively. And so our company called Ginvenate, John is a member, joined the entrepreneurship program at HKUST, and it is now a member of this program. So GVN is a biotech company engaged in the research and development, production, and commercialization of skincare and healthcare products. And we make use of microbial expression system. The one that I talk about, E. coli, has been very useful for the production of EGF, but for another interesting protein, basic fibroblast growth factor, which is important for the formation, production of collagen as well as elastin in the dermis, then we find that actually EGF, E. coli could not be able to use as a suitable host. Then now in our laboratory, we have another system. We engineer B. subtilis, Bacillus subtilis, and we have found that B. subtilis has been a very good candidate to produce good quality BFGF. As Chris introduced, of course, I mean, we make use of EGF to produce skincare products, and so far, in these 2 years, our company has been able to produce 2 lines of cosmetic sample products. One called the Gene line, comprising 9 products, and then the other one is Sabio. Now, Sabio is a new product, and these products will be out, at the end of this month. And this is in the collaboration with beauty salon in Hong Kong. And they are very interested in our technology, and they are very confident in our technology also. And so just very recently, we have established collaboration to produce this new line of product. Now, most importantly, we also develop a couple of healthcare products called the Gene Care product. Now, they contain much higher percentage of epidermal growth factor. And we find that using higher dosage of HEGF, then the EGF can exercise, surprising curing effects on hard-to-heal wounds. Now, the first one, we started our study in collaboration with a local hospital, the United Christian Hospital, in the year 2000. We made use of EGF at a concentration of 0.04%. And we find out that with this high percentage of EGF, as you know that this is 0.04%, and the one that we use for cosmetic application is 100-fold lower. So no wonder, people in the previously, they said that EGF was not effective for the healing of diabetic foot ulcer because they used very low concentration of HEGF. Now, in this study, we find out actually there is a so-called dosage effect, and we carry out a double-blind placebo-controlled experiment. We find out that EGF at this dose, 0.04%, actually is very effective in healing diabetic foot ulcers. Now, as shown in this survival plot, actually those people treated with 0.04%, the healing rate is very significant. Whereas the one treated with 0.02% is basically the same as the placebo, and it is not significant. So the conclusion in this study is that only if we use HEGF higher than 0.02%, then they will exercise the effective effect on the healing of diabetic foot ulcer. And also that 95% of the patients that tested in the study, their diabetic foot wounds got healed in about an average time of 6 weeks. Now, when our results compare with another growth factor, platelet-derived growth factor, PDGF. Now, PDGF was previously approved by FDA as an agent for the treatment of diabetic foot ulcer. When you look at this figure, now PDGF, not only that it is more expensive than EGF, it is about 5 to 10 times more expensive than EGF. But when you look at the healing rate, you could see that it's only 50% when they use 100 microgram per gram of PDGF, when compared to placebo. Placebo is already 35%. But anyway, we find out that HEGF is much more effective than PDGF, and also it is much cheaper, So even though nowadays, EGF is not classified as a drug yet, but locally our hospital have already started using EGF to treat diabetic foot ulcer. Now, the other hard-to-heal wound is bed sores, and it occurs in a lot of elderly individuals, particularly those, bedridden, those lack of mobility. And these ulcers tend to develop just right above the anal region. Now, this is a very serious case, and we will approach, and finally, we make use of 0.04% of EGF to treat this. And in about 10 to 15 weeks or so, the wound was treated readily and healed finally in about 15 weeks' time. And in 2003, we applied for PCT application, and recently we have been informed that we are close to the very last stage of being granted this patent. Now the next one is an acute allergy. Actually, this one is allergic to drug. This particular case is called Stevens-Johnson syndrome, and this gentleman, he somehow took some pill, and then he was suffering acute hypersensitivity. And then you can see that the hives develop on the skin and finally ruptured. And then you could see that they are actually broken parts of the skin. And he was sent to the ICU immediately, and then he was treated with 0.04% of EGF. And surprisingly, in 2 weeks' time, for example, the wounds on the face were healed readily. And we also published it on Dermline, Dermatology Online Journal in 2004. Now, the next one is serious burns. Now, this lady, the calf region, actually was scalded by hot water while she was ironing, and she went to see doctor, and of course the blisters burst, and she was just given some antibiotic, and she knew that we had this 0.04% cream, so she, obtained some from our company, and then in 5 days' time, basically you could see that that's the skin, was very, rejuvenated very nicely. And another serious one is about treatment of a surgical wound. This particular person, he is diabetic, and he received a bypass surgery. And somehow the vein was taken from his left thigh, and after a month or so, it was not healed. And you can see that already gangrene development. And he actually lives in Vancouver, and he knew that we had this cream available for the treatment of hard to heal wounds. So he approached us and then obtained some of the cream. And after 16 days of treatment or so, the wound healed readily. And then we were very surprised that the wound actually healed without any formation of very ugly looking scar, right? And even in 2 months' time. And last of all is the treatment of psoriasis. I mean, psoriasis occurs very commonly in Caucasians, and this we undertook this study in collaboration with a German doctor, and actually he used our cream, and then he did the study in Germany, and he found out that actually EGF is very effective in the treatment of also psoriasis. And as GVN is a new company, and our model is to form as many collaborations as possible with different parties. So if you are interested in forming collaboration with us, please contact me. Lastly, thank you very much.